Chris, you want to get us started?
Yes. Okay. So welcome everybody to the Biocomplexity Institute initiative exploring biocomplexity distinguished speaker series. And obviously, because you're here, you know Michael Waterman is our speaker today. He is distinguished institute professor in the Biocomplexity Institute, and his obviously his longstanding position at USC as an emeritus professor now is um, something that we all know him as, and we have for many years uh, been amazed by his work. So he's an early pioneer in bioinformatics, one of the most important thinkers in computational biology, and one of these people that actually has written a couple of papers that have gone through the paradox of at some point they're so ubiquitous that you don't cite them anymore. It's a pretty amazing reality. But all of modern genomics owes an enormous debt of history to Michael Waterman's work. I'd like to spend just a couple of minutes, talking about connections between this and the Biocomplexity Institute, and the relationship between mathematics and computing and information as a topic, as maybe the unifying topic in whatever we might call biocomplexity, and how it's related to Mike and our relationship with him. He comes to us primarily through a connection with Christian Reidas, who is a professor here in the Institute and a mathematician and a professor in the math department. Christian's work for a very long time has had to do with RNA, sequence to structure, secondary structure properties. And he's connected in the course of that work and his background with Michael. Christian and our organization came when we were at Los Alamos. All of us were at Los Alamos. Christian was at Los Alamos. I was at Los Alamos. Others in the organization were in Los Alamos. But the issue of how this simple seeming thing of sequences of finite alphabets and just asking questions about their differences and stuff, how tortuously difficult this is and how ubiquitous it is, this simple seeming thing, frames a connection that goes back, if we just look at Los Alamos, to a very, very interesting history. Christian and I connected through Giancarlo Roda, a very interesting line of work at Los Alamos that was viewed as a little extreme and had to be reviewed. And Giancarlo was our reviewer. Now, if you know anything about him, you know that it's not always a good thing if he becomes your reviewer or did become your reviewer, but it was an incredibly important and interesting thing that he did that. There's other people there, Bill Byers, Giancarlo Roda, others, that we all are connected through, because Michael Waterman also was at Los Alamos about 15 years before we were. So there's a really, really curious and interesting connection through this intellectual history that continues to be woven. And one of the values of the Biocomplexity, so one of the values of these relationships, is that these intellectual lines live through the continuation of these relationships. And it turns out that our own Dick Beckman in the Institute, one of our honorary members here, but he's been with us for years, was also at Los Alamos. He studied there at the time that he was around Michael Waterman. He was studying sparse designs, I believe, statistical designs of computer experiments. Which is also a fundamentally important problem, and they knew each other. And Temple Smith, who will speak today, was also there. So there's this incredibly interesting, deep, important relationship between these lines of work, and they've converged here, and we are working again together, or working actually for the first time together, but this work is again converging together, and our connection through Christian Rightus, I think, is really very well served. But Michael, I don't know how to thank you enough to work with us and to give this talk today. It's a wonderful privilege to have you here, and we all greatly appreciate it. Welcome, and I hope you enjoy the day. So we'll go now to Kevin Skadron, the chairman of the computer science department, for some remarks.
Hi, everyone. I'll keep it very brief. I also want to join in welcoming Mike Waterman. It's really a privilege to have him here at UVA and to have this event today. Mike, as Chris mentioned, is one of the founders of the field of computational biology. He actually got his PhD in statistics and probability back in 1969 at Michigan State. Back then, computer science was just in the infancy of coalescing as a field, and there were relatively few places that even offered PhDs, but it was also a time of a lot of ferment as a lot of ideas were coming together in both the areas of computing and biology. And as Chris mentioned, Mike went on and spent much of his career at USC, actually becoming university professor there in recognition of his wide-ranging and high-impact contributions that really spanned conventional disciplinary boundaries. I mean, he's really an exemplar of interdisciplinary work. And, you know, he's probably best known for Smith-Waterman. This algorithm is still widely used and of high importance. And we'll hear from Temple Smith momentarily. But I mean, you know, Mike's contributions are so broad. So just as one other example, the Eulerian-de Bruyn sequence assembly algorithm is now widely used in next generation sequencing. So once the reference genome was developed by the Human Genome Project, sequencing a new human genome became much easier because you had a reference that could provide you with hints on where your fragments of DNA actually belong. But when you're sequencing a new organism or a new virus that hasn't been seen before, like perhaps a novel coronavirus or some other DNA that you discover that you're trying to figure out what the heck it even is, the challenge is much greater. And that's where an algorithm like the de Bruyn's sequence assembly algorithm comes into play. Actually, my group is working with this now in the context of trying to make it faster with hardware acceleration. So, you know, yet another example of the many ways in which Mike's work continues to be highly important and beneficial to society. And of course, you know, he's been widely honored with a variety of recognitions, multiple honorary doctorates, the Dan David Prize in 2015. He's a member of the National Academy of Science, the National Academy of Engineering, the American Academy of Arts and Sciences, multiple international academies. And, you know, in the interest of time, I'll stop there, but he recently joined UVA and we're so lucky to have him here. And with that, I'll pass the sort of virtual microphone over to Christian.
Yeah, thank you, Kevin.
Great
so
to have
as for academies, I have to pick up a particular one, the French Academy of Science. So it is a particular privilege for American scientists to be awarded that. So, but to get to my more formal introduction, it's my privilege to welcome you, Mike, here as a speaker for our Exploring Biocomplexity event. And I can say that I know Mike for decades now. We started, you know, him inviting me to USC, giving a talk, and over the course of the years we became collaborators, colleagues, and eventually friends. So right now he's a member of the mathematical biocomplexity group at the Institute, And he is still active and an integral part for our work on interpreting genetic data. So it's already clear from what Kevin said that he has made epic contributions in the field of bioinformatics. If not, he has shaped the field. And from mathematics alone, he has contributions from probability theory, statistics, where he originated from, to combinatorics. He has a particular beautiful bijection between linearly ordered trees and secondary structures that gives really beautiful formulae. And finally, even to topology, right? Notably algebraic topology. So here's a construction together with Penna about certain simplicial complexes arising from RNA secondary structures forming a high-dimensional sphere. So it's a very structural result, and you seldomly see this kind of range of people solving problems and also forming theory, you know, incarnated in one person. So it's truly remarkable. So I personally find his publications super clear and beautiful to read. I particularly like his lectures. It's a shame we can't see him on the blackboard. So we have to invite him as soon as we can after this pandemic has calmed down to give a blackboard talk, because this is really where you can see much more of Mike than in a PDF slide talk. Now, I just want to point out one particular issue, since Chris brought up Giancarlo Rota. So Rota is quite famous for being an interesting character, truly unique mathematician. That's for starters, we all know that. But he also was, he made a quote about mathematics and biology in particular, which I want to read to you. So the lack of real contact between mathematics and biology is either a tragedy, a scandal, or a challenge. It's hard to decide which. And in that, I can only conclude that Mike found an answer to this. And I guess if you find an answer to this challenge, then success is really yours. Because Mike is truly a mathematician who succeeded solving problems that are of utmost relevancy. And for mathematicians, this is a gigantic challenge, and Giancarlo hit the nail on the head on that. So Mike, we're happy to have you as a distinguished speaker. And before I let you start, I want to give the microphone to Temple Smith, who has a recorded brief introduction.
Well, Mike Waterman and I met in 1974 in New Mexico at Los Alamos at the National Lab. And it was an interesting meeting. In fact, Mike has written a chapter in one of his books about the day he and I met. And it's not necessarily a compliment, but I'm sure he meant it that way. We were speaking two different languages to some extent. Mike was approaching things from the point of view of sort of pure or serious mathematics. And I had just gotten involved in molecular biology. So I'm speaking molecular biology as viewed by a nuclear physicist, which was my background. So it was, as he and other people at that time pointed out, it was an interesting conversation, to say the least. And it was that work that summer that eventually led to our trying to get an alignment system that would identify local sequences similar between two. And that was partly motivated by the fact that relatively recently, it was found that genes were in pieces. They were not in a single piece. And therefore, trying to do a global alignment, which other people had done by various methods, wasn't going to do what we thought needed to be done. So that was the motivation behind it. But the story I want to point to is another time when I was on sabbatical at Yale, and this would have been 79 maybe. And I was at Yale trying to become a real biologist for a year. And Mike Waterman was visiting East and he came by. And we had lots of discussions and we decided to go to lunch. And as we walked to lunch, we took a shortcut through the geology building. And as we went through the lobby of the geology building, we both sort of froze in our tracks. In the middle of the lobby were two columns, strata columns. And somebody had taken ribbons and tried to connect similar strata on the two columns to each other with ribbons. They were doing a sequence alignment. Well, Mike and I went off to lunch. We didn't eat much lunch. And we finally decided, look, I don't know why we need to publish this paper, but it hadn't been published yet. Because the geologists apparently have already solved this problem. So we walked back, but Mike being Mike, as we walked back, we went back through the geology building and he says, I'm going to go in and ask them what algorithm they use. So he went into the department chairman's office and came out with a grin you couldn't believe. He says, they don't know how to do it. They've never done it mathematically. In the next 2 days, Mike and I wrote our first geology paper, my only geology paper. And to me, that represented one of the key aspects of science and our work together. And that is, you could reach into some other field and some other area and recognize to apply a problem to this new area. And that's one of the keys to good science. And it is interesting that Mike went on and over the next couple of years and wrote a couple more geology papers, mainly because the geology problem is a little different than aligning DNA or two proteins, because when you align two proteins, you take alanine and align it with alanine, or you don't. But in geology, if you have a layer of coal, yes, you can align it with a layer of coal, but it could be 5 times bigger or much thinner. So it was like trying to align the amino acid alanine, a skinny one, with a fat one. So the problem was different. And I think over the next couple of years, Mike published at least 3 papers on expanding our original algorithm to problems in geology. So that is the way I still think about our relationship. And we have worked together and talked to each other probably over the last, I don't know, 30 years, I guess. It's hard for me to count that far. So thank you.
All right, so at this point, Mike, you have the floor to give your talk.
Okay, I think this has to link to my screen, I guess, right?
Yes, hopefully it will.
Oh, share screen. There it goes. Are you seeing my screen now?
Mm-hmm.
Great. Okay. So if you read this title, I suspect you're thinking, oh my God, there'll be case study number 2 someday. Anyway, we'll start with this one. It was after I was an undergraduate, I saw this amazing phrase from Eugene Wigner, the unreasonable effectiveness of mathematics in the natural sciences. And for somebody who who did math and dabbled around in philosophy, this is just quite an amazing sentence. How does math relate to the real world? Why is math useful at all? How does mathematics that seems not of our world turn out to be useful in understanding physics, for example? And there's a whole Wikipedia page for this this phrase, and it's just wonderful. And I want to say the examples from physics include mechanics and string theory. It's really all over the place that math and physics are very linked. And in Victor's paper, he says, the miracle of the appropriateness of the language of mathematics for the formulation of the laws of physics is a wonderful gift which we neither understand nor deserve. And then he goes on. A much more difficult and confusing situation would arise if we could someday establish a theory of the phenomena of consciousness or of biology, which would be as coherent and convincing as our present theories of the inanimate world. And for sure, it's more difficult, but I'm not sure why it would be confusing if we had a theory of consciousness or biology. And is this miracle going to happen between math and biology? Will it ever happen? Or maybe it's already happened. And that's really what this talk is about. If you go to Wigner webpage, there's a bunch of people quoted. And one is my friend Len Edelman from USC. He's the A in RSA encryption. If you've heard of DNA computing, he invented it. And he got into this biology connections after I did, but it's worth reading this quote. Sciences reach a point where they become mathematized, and the central issues in the field become sufficiently understood that they can be thought about mathematically. The field was undergoing a revolution and was rapidly acquiring the depth and power previously associated exclusively with the physical sciences. And here's the key. Biology was now the study of information stored in DNA, strings of 4 letters, ATGC, and the transformations that information undergoes in the cell. There was math here. Now, there's another view quoted on the same page by the famous Russian mathematician Israel Gelfand. There's only one thing which is more unreasonable than the unreasonable effectiveness of mathematics and physics, and this is the unreasonable ineffectiveness of mathematics and biology. I knew him toward the end of his life, and he tried very hard to have an impact in biology and found it very frustrating. He was really one of the great pure mathematicians of the 20th century, and I could resist sharing this with you. There are 3 people known as Big Gelfand, Middle Gelfand, and Little Gelfand. And Big Gelfand, of course, is the grandfather Israel. Sergei Gelfand is a distinguished mathematician, works for the American Math Society. And Little Gelfand is Misha Gelfand, who works in bioinformatics. And I think I screwed up here. There we go. There we go. And he's a rector at a university in Russia, and this guy is so brave. He criticized the government. He's not put in jail as far as I know, but he's somebody to know about. And for sure, many of us started in this area, but this Len Edelman point of view. And I've seen waves of people come in from computer science, information theory, statistics, math, physics, motivated by that kind of pure math view. And these people often said, just tell me the top 10 unsolved problems that you're not smart enough to solve. But in fact, anybody who moved very far down this road found biological reality sort of didn't allow that kind of approach, but it was very exciting at the beginning. For sure, there was mathematics and population genetics. The Hardy-Weinberg law in 1908 Hardy, who bragged that he'd never done anything useful, is today known because of the Hardy-Weinberg law. Now, Mendel's paper was in the 1860s, but it wasn't noticed again until 1900. And in the 20s and 30s, Fisher, Haldane, and Wright laid a foundation that integrated natural selection, Mendelian genetics, and there was serious math in their work, no question about it. But they did not know physically what a gene was. So they were trying to understand inheritance, but they didn't have, the molecular microscope that we do today. So I think biology was slow in sophisticated developments like we have now, and lagging behind physics and chemistry because we didn't have the technology that would allow us to discover the double helix. So we didn't even know what a gene was written on or how it was written. Sequencing DNA didn't happen until the mid-1970s. The Human Genome Project didn't begin until 1990. And I think from my point of view, biotechnology and computer science had to be there before we could make deep progress into human biology. But while there's no grand theory, the mathematical and computational sciences are essential to biology now. And I'll concentrate the rest of the talk on genetic genome database search. So just how do you find stuff that's relevant and important? And the DNA sequencing that started in the mid-1970s, the first draft of the human genome was 2001. And our database, GenBank, holds 6.25 trillion base pairs, 1.6 billion different sequences from almost 500,000 species. And the cost of sequencing during the Human Genome Project declined, but maybe by a factor of 10. So the Human Genome Project optimized Sanger sequencing, the 1970s method, but it was still Sanger sequencing. And then things right after the Human Genome Project, new technologies came in and things really boosted. Here's the cost of a megabase of DNA sequence, just one sequence read, goes in 2001 from $10,000 to about 10 cents in 2020. So that's what 4 factor of 10 orders of magnitude decrease. So the cost and the speed is gone. It's just the cost gone down, the speed's gone up, and we're just swimming in an immense amount of data, some of which we don't understand too well. Now, behind all this, and what makes algorithms like Temple's and mine work, is that there are common patterns of sequence within individual genomes and between different genomes that have been kept around because the function of those sequences are important and are preserved. So you can see cytochrome C in bacteria and human. And I think the intellectual foundation of what we do really comes from Darwin's insights about natural selection. And so here's sort of some of how that works. How does sequence evolve? Letters change, mutations of nucleic bases or amino acids. A sequence can be added, a sequence can be chopped out, a sequence can be picked up and turned around backwards. It can be moved somewhere else in a genome, and it can be duplicated. All these are important, and we will focus on single letter changes and single letters that are inserted and deleted. It's enough to make it challenging. So there's something called sequence alignment, and this is my favorite thing to say. What are we doing and why are we doing it? We're interested in the relationships between the word what and why. And here we've aligned them up where we have the W's and the H's aligned across each other. And here we might think the A was added in the WHT sequence or erased from the WHY sequence, and the T and Y somehow had a common evolutionary ancestor. And to make this computer science, go back here, we have scores for these, and most of this talk, we're gonna just count identical letters, but you can have a complicated metric for this. And letters like T over Y, you get a negative score, and the indel letters, you get negative scores. So that alignment scores 2 identities, 1 mismatch, and 1 indel. And the local alignment problem Temple was talking about, which kept us scratching our to-be bald heads for years, was finding in a long genome sequence X and another long genome sequence Y, find the pieces I and J that are most alike one another. So we're really looking for global optima, but they're local phenomena. And so global alignment is just what I just showed you, these 2 sequences, and this score is the maximum score of all possible alignments. And the number of alignments grows exponentially, as you probably would easily guess. And there's these dynamic programming algorithms that Temple and I worked on that reduces the exponential problem to polynomial. Local alignment is looking for these 2 intervals, looking at the global alignment score of those 2 intervals, and then looking over all possible choices of those intervals. And our algorithm in its simplest form is here. We don't need to go through it, but it's a quadratic recursive algorithm. Now, and so go back here. We started working on this. We did distance between sequences at the beginning. So it would be a D instead of an S. And we felt very strongly, being mathematicians, that metric spaces are what we want. We're constructing metric spaces. We'll get the power of mathematics here. But in fact, this algorithm is a similarity algorithm. It's not a minimum, it's a maximum thing, trying to accumulate positive stuff. And the reason for that is that it's very hard to tell from distance what you actually got the distance from. For example, AA is identical to AA, so it would have distance 0. And however many of these are, 15 A's in a row have distance 0 from 15 A's in a row. You can't tell the difference between those 2 zeros. But if you did similarity and you were counting up identities in the matches, there would be 2 identities here, and there would be 15 in the other one. So you very definitely can tell the difference between 2 and 15. And so that's the reason similarity gets used. And there's a story of how we actually discovered that for every global similarity algorithm, there's a dual distance algorithm. So it was how our mind was in this. So what's a scientific math computer science problem? And local alignment is what we want to do in genetic database searching. And the algorithm I just told you, our dynamic programming, is too expensive in space and too expensive in time. So we need efficient computation, and we have to evaluate what we find. So computer science and statistics. And in fact, statistics in this case guides the practical solution for the efficient computation. I'll tell you about that. So computer science is a serious subject since I was an undergraduate and heard there was something called a computer. We can do comparisons using suffix arrays, suffix trees. There's these beautiful Burrow-Wheeler transformations. They're studying with seeds and space seeds. There's locality sensitive hashing. And probably the most of these sophisticated approaches, probably the most effective one is Ming Lee, who's in Toronto, or in Canada anyway, has a pattern hunter program that really does this. But what any biologist will tell you is that when they do database searches, they use BLAST. BLAST was published in 1990. And what it does is the following. It takes a query sequence that you want to learn relationships with database sequences. And it takes that sequence before it does the search, and it finds all pieces that have statistically significant matchings. That'll be a long list, maybe a few 100,000 of those matchings. If they were in the database, you would say, I'm interested. Then you search the database for those exact matchings. When you find them, then you do restricted Smith-Waterman in the neighborhood, trying to improve the alignment. And they have to emphasize statistical significance of the hits. Blast is a verb. We'll say, just take that sequence and what did you learn when you blasted it? And blast is a sophisticated heuristic for Smith-Waterman. It's cited 75, 80,000 times. It was the most cited paper in science, I think, in the 1990s. And it's essential for genome biology. So this is an example of where mathematics and mathematical approaches deeply affects the progress of science. And what about the statistics part of this? So if you just put down 2 random sequences and you looked at the largest number of matching letters, the score would grow proportional to the length of the sequences. But if you were looking for the longest exact match between those sequences, it grows proportional to the logarithm of n. Yeah, and sorry, here's an example. Here's 2 random sequences, I guess, of length 20. And where you can see here, when the letters are identical, you get a 1. When the letters are not identical, you get 0. Add these all up and you get 10. And since I constructed this with probably the A and B each had probably 1/2, that's the expectation. But if you allowed free insertions and deletions, you can increase that score a lot. If you go down here where we put spaces in some of the sequences, we get 15 letters matching. And notice that it's 50% increase by being able to put these spaces. There we go. So the study of this longest common sequence problem, the score for the last one, started in 1975. Svadl and Sankoff, a brilliant paper. They proved that the matching score grew as a fraction of n. And if you don't allow insertions deletions, that fraction is 1/2. If you allow insertions and deletions, it's 0.82. It increases more than 50% on average. And with subaggregatic theory, you can prove this. They did it a different and harder way. But it's been 45 years and we still don't know what alpha is in the simplest case. I would love, and I have a conjecture that the variance for the longest common subsequence score is proportional to sequence length. Conjecture before it was into the 2/3. There's a few proofs, but this is understanding longest common sequence statistics is far beyond our abilities. But local matches, in fact, is something else. So I'll try to do this with a sequence of coin tosses. Here's our heads and tails. There we go. Now, we're going to look at success runs. So how about a run of 3 heads? So here's a run of 3 heads, overlaps another run of 3 heads, and there's another one here. So there are 3 runs of 3 heads. But if you looked at clumps where there were 3 or more heads in a row, you get 2, because this one is the overlapping aspect of this is part of where the cool math comes in. So you cannot apply the Poisson distribution to counting clumps, but you can apply it to the Poisson distribution to counting clumps that has at least k heads. And so the mean of that would be normally you'd have to start with a tail, you'd have to get at least k heads, and then you're home free. So there are essentially n possibilities for that. Now, this is a cool part of this story, I think. I don't know how many people in the audience have heard of someone named Paul Erdős. there's a wonderful book about this character titled The Man Who Loved Only Numbers. And however you take that, it's right. Paul is an extremely famous guy. He died of 1996. He had over 1,500 papers, over 500 collaborators. A lot of his work is discrete math and combinatorics. And there's a number called your Erdős number. So that's the length of the shortest collaboration path to Paul. If you publish with Paul, your number is 1. He was famous for taking basically speed. And he had a bet with Ron Graham that he could go a month without taking any of those kinds of drugs. And Ron, he managed it a month and took his $500, and then he went back on the drugs because he had some calculation of how many theorems weren't proved because he wasn't taking speed. I love it. He had no home. He traveled around the world, visiting math departments and institutes, got money from awards and stipends. Ron Graham filled out his tax forms. I mean, this guy was, in some sense, hopeless in some sense, and truly a genius. So in 1982, at Los Alamos, there was a computer called the Cray, and we ran our alignment algorithm on all pairs of known DNA sequences. And we wanted to learn new biology from the alignments, but you need to know what the background distribution is so you can distinguish the important stuff. And there was a conjecture from this data that I wasn't too convinced by, that the score grew like log n, as I've told you is the case. The evidence was weak and it didn't have any theory. But we did this comparison. We wrote a paper and it was almost impossible to publish it. We submitted it and we rejected, and we submitted it, was rejected. And it was absolutely the edge of what was possible to do computationally. The Cray was a vector machine, and Temple did some clever thing to make the algorithm work. There we were. And this is a picture of the Cray. It looks a little fuzzy, but I always thought it looked like it should be in the lobby of a Holiday Inn somewhere. It's just possible to believe this was the world's most powerful computer at the time, but there was one at Los Alamos. And when I went to USC, in the library, found a paper about the largest rectangle of ones, an n by n rectangle of random zeros and ones. And the answer had to do with the logarithm of n, and my eyes lit up. It referred to something called the Erdős-Rényi law, which was about long head-ridge runs and coin toss. And the famous Mark Katz, the Feynman-Katz formula, brought me to USC and was there and said, the next day after I found this, tomorrow my friend Erdős is coming to lunch, you be here. Well, I showed up. And of course, there was too much talk at lunch for Erdős and I to get down to business. So he said, come over to Caltech, we'll have lunch at the Athenaeum. And even at the Athenaeum, there were lots of people. So eventually we had to go to his room. So I go to the room with Erdős and explain the problem to him. We talk for a while and he starts looking a little vague, and then he just goes to sleep on me, slumps down on the bed. His head is on the pillow. And I'm sitting there, and it's not a fancy room, but I'm sitting there thinking, what did you ever do to deserve being in this room with Paul Erdős? And maybe 10 or 15 minutes, I thought, well, I'll just leave this old guy to his sleep. And I got up to leave, and he shook himself and said, no, I'm not sleeping, I'm thinking. And so the results of his thought were that it was possible to solve this problem, and it sort of left it there. And I was sort of too shy, I guess, to suggest working together. If we'd pursued this together, my Erdős number would be 1 instead of 2. But But at any rate, during that year, Richard Arati and I got somewhere on this problem. And I think I could at least indicate what the intuition is. And of course, successive head runs, runs of black at the roulette wheel or something. This is always interesting to people. And so what's the longest head run on a random sequence? And so it's possible to get a little bit of work for this. So imagine the longest one, there should be about one of those. So let's let one equal the expectation of how many of such head runs there are. And such head runs will start with a tail. There'll be k head runs and there are n places to start it. So if you solve this problem for k, you'll get essentially the logarithm to 1 over the probability of heads of n. So this is not math, but it gives intuition. And the year later, Paul Erdős came back a year later, and I told him this answer for our answer for sequence alignment. Well, he, of course, knew this for coin tossing. And he was surprised, which was almost better than having an Erdős number of 1. So between two sequences, it's kind of the same principle. How many places are there to start the matchings? And then what's the logarithm of that number of places? So there are n squared places. And if you remember what to do with logarithms of powers, this becomes twice our familiar log base 1 over p of n. So this requires a technical proof, but it's the same sort of declumping Poisson ideas that we had in a single sequence. And we've done a lot of work on in this direction. But one of the ones that's top of my own list is a paper Richard Arati and I wrote, published in 1994, but we spent years at this. I discovered it by a simulation. And the simulation was, if we don't penalize for insertion, we penalize barely or not at all for insertion deletions, the local alignment score grows with n. If we penalize seriously, like don't allow any, the scores grows with log n. So I assumed this plot would have n in one corner, log n in another, and be mush in the middle. But in fact, the transition from linear to logarithmic was abrupt. There's a line through the parameter space. On one side of the line, it's linear. On the other side of the line, it's logarithmic. And the line is determined by solving when the fraction of score growing with sequence length was 0. Anyway, Richard and I spent years on this, and finally, the first proof was head splitting, but at any rate, I think it's readable now. And what about this? So all this is getting around statistics of the search. Statistical database search, my version is Poisson approximation, but in fact, that's not the formulation that the BLAST connected stuff. So there's something called the Carlin-Altschul distribution, published not in BLAST in 1990. It's a basis of statistical analysis, and it wasn't until 1995 that Dembo and Carlin provided a mathematical proof. Carlin was a genius. He wrote over 500 papers, he had 10 books. He worked, and if you just read this, game theory, analysis, math stat, total positivity, probability, random processes, math economics, et cetera, et cetera. And he used to say when somebody would ask him something as tacit processes, and he would sort of snarl at them, "It's in my book." So here's a picture of Sam. He was a serious competitor and raised the level of discourse in this subject. So given that, what about more details? What about the algorithmic aspects of BLAST searches? So Gene Myers, who's a BLAST co-author, had a very sophisticated, deep string matching scheme, but it got shunted aside in the importance of making a practical a program that would satisfy biologists and so on. So So you remind you, we have a query sequence, we have a large database of previously determined sequences, and we want to find all statistically significant matches of the query sequences to any of the database sequence. So as I said before, look at intervals, which could have significant matches a la this Poisson approximation. And then after you have all such matches, you could search the database. So at this point, what's happening next? BLAST is an essential tool for modern biology, but what do we do about the huge increases in the volume of data that comes out? Just think about how much data has been produced by sequencing COVID viral samples. So Bonnie Berger at MIT and her former student, Bill Yu, have approached this problem using information theory. And I had tried something in this direction once and didn't get anywhere. I'm really impressed with this work. So here, so I'll conclude with these 3 slides from Bonnie. Here we've got raw bases that are sequenced per dollar. So you get more and more sequence per dollar as time goes along. And this is, this is computing power on the right, just raw computing power. So genomic data is rapidly going off the chart. Computing power is increasing, but not as fast as the data is increasing, and disk storage is even more a problem. So some point in the fairly near future, computers won't be fast enough. What are we going to do? And I don't know whether this is a Star Trek or Lost in Space picture, but here we're blasting through space. So you really need to do something that searches the data sublinearly. And their idea is to take the set of existing sequences and look at their statistical structure. How are they positioned in sequence space to speed up the search, and so narrow the search down to regions where you will find good matchings, and not do searching where it's irrelevant. So this was done something called metric entropy, which has a bunch of covering spheres in sequence space. And it ends up the biological data has lower fractional dimension, so that the search can be done much more efficiently. And I think, yes, got a slide here. This is runtime in seconds and the number of genomes that you're searching. And BLAT is one of the modifications of BLAST to help it run even faster. So this is a BLAT running, and this is the data, Bonnie and Bill's algorithm, the compressive database, and they have an even faster version that's not quite as accurate. But the compressive version has very high sensitivity on real data, so it's very cool. It doesn't, I think their work doesn't get universal use. Kind of the reason why new word processing algorithms don't get as much use as Microsoft. When you're in there first and it's big, you're kind of stopped. So anyway, this is sort of a guided tour over the top of sequence search in biology, and it's certainly now that we've read the code, as Edelman put it, we need to understand how organisms operate. And this is a much harder problem. So I guess my title here is, we're not unreasonably effective, but we're sometimes effective on some things. The new title, Systems Biology, Quantitative Biology, really wants to understand how biology really works. And just to give you a hint of how hard that is, 80% of the explanation, quote unquote, for a person's height is determined by DNA sequence variants that they inherited from their ancestors. We have 700 gene variants that affect height. And that's not the whole story. I read another paper where there were 5,000 associated genes that were supposedly affecting height. And it's extremely hard to make sense out of that. And when you take that genetic complication and you add in the environment complication, the other 20%, say, this kind of simple, easy to understand human trait is not going to be understood very deeply for a long, long time. And I wanted the Robert Frost quote to end on, we have miles to go before we sleep, but it's sort of, we probably have centuries to go before we understand biology. But we're started, and it's an exciting time. And thanks a lot for listening.
Yeah, thank you, Mike. So I'm gonna move on to the Q&A section, and I will take a little bit of time to pose a few questions with respect to your talk and also what Temple said. So Temple mentioned briefly that if you align strata, you actually have a scalar associated with the sites to be aligned. And he mentioned that you had a few geology papers subsequently. I'm wondering, how did you deal with that?
Yeah, so I even went to, let's see, I'm trying to figure out how to get my Zoom back full screen. How about that? Nope. Oh well. Anyway, so I even went to a school on well logging, which is, when the Schlumberger puts the instruments down the well hole, measuring resistivity. So that would be matching signals, which of course is a topic related to alignment. The key thing that I really ended up working on was, unlike sequences, if you have one group of geologists determine where the strata boundaries are and what the strata are and what they're named, another biologist may, on the other side of the gorge, do that himself, and some of them will put several. What the guy on my left made 3 different strata, the one on the right will make them into one strata. Mm-hmm. So there was the algorithm I did with a geologist at Los Alamos was sort of fusing strata together to try to do the best matching when you don't know what to fuse ahead of time. So it was a lot of fun talking about talking with the geologists. I learned that that it's the types of rocks and stuff are related by the size of the particles making them up, and even when you do screenings, like when you take gravel from a stream and you're trying to see what the composition of the gravel are, the mesh, they have coarse mesh and increasingly finer mesh and layers, and they shake it, and those mesh sizes have to do with a statistical distribution of how rocks turn into sand by random breakage.
So I'm wondering if you look at your recursion, where eventually you apply that score, you have this plus S ij term appearing in the recursion when you try to do the local matchings, right? And I'm kind of wondering, when you put a weight on the sites, which say 5 tons of coal versus 1 ton of coal or something like that. So does that go into your scoring function, or what? Is that a deeper structural change for the algorithm, or is it just, can this all be remedied by changing the scoring itself?
I mean, it is sort of like biology. It's all wrapped up together. How you do the relative scoring depends on your view of geology, I guess.
Right. So you're saying you would put then the weights somehow into the scoring function themselves when you do the recursion.
Yeah.
Hmm. So on a more general from a more general point of view, so bioinformatics, I mean, 10 years from now, what do you think is going to happen?
It'll get more and more.
Does it feel
it's going to get more and
like?
more.
I mean, yes, you kind of came to the point where you say, well, I mean, we produce much more data than our computer growth is able to handle, let alone disk space. So apparently then the logic is we got to deal with the data more smartly.
But
right.
There are two aspects. I mean, when I talk about that database growing, that's back to the mathematician sequencer's view of the data, right? That's not, it's not about the real complexities, which is why are you the height you are? What genetic determinants in your genome caused you to be as tall as you are? And that's a much harder question.
So what you envision is happening? Are we gonna, plow along and see this increase in data accumulation versus computational power, and you think there are conceptual changes on the horizon? What is needed in order to, I guess, interpret the genetic data in a more efficient way?
Well, it's somewhat, yeah, I mean, of course, we're interested really in what happens in real cells and real organisms, and that's we take, what was that old joke about somebody find their keys, who lost their keys by the road at night, and they're looking under the only lamp that's around, and somebody says, "Did you lose your keys here?" No, but why are you looking here? Because the light's good. And that's all we can do is the tools we've got and push it out as much as we can.
So I want to get back to Rother's quote about this tragedy, scandal, or so what do you think? I mean, are we better? Are we improving that challenge?
Yeah, I really think so. I mean, I have to tell you, wrote a story. He wanted to write a paper with me explaining biology to mathematicians, and I was too busy. So funny, after a while, and he was visiting USC at some time, I've got enough. Okay, I've got enough time now. I told him, okay, we can start that paper now. He said, no, we can't write that paper. Why not? You know too much, he said. But by which he meant I'd gotten too far into biology to make it, to abstract it into a coherent.
So let's say for someone in mathematics who wants to be successful in biology, right? So maybe I can extract something very useful for anybody in that category listening to us right now. You have a particular advice? I mean, what made your work so successful as opposed to others who try to make an impact and apparently don't get their, feet, so to speak, have traction?
I guess for my own self, I mean, Temple said, he was the translator of biology to me, which is, as he indicated, a little scary, but when I finally decided this is what I was going to do, after I left Los Alamos, I went to UCSF, the biochemistry and biophysics department. I went to every talk, every biology talk that was given, and I tried to learn biology. And after that, I didn't work much with Temple after that, because I sort of was getting stuff from the source. And I think you just can't come into this and not take courses in biology and work with biologists at this point. It's too complicated. We were able to do it at more of a distance, but it was a different time.
Yeah, so I know from personal connections that there are several combinatorialists who went into protein folding, and they went in and said, we're going to solve protein folding in a similar vein, and that certainly didn't work out so well. But it seems that what Rota was specifically referring to, you either have the ones that apply mathematics but not really do new mathematics, or you have the ones who do mathematics and are not really so much interested in biology because it becomes messy so quickly, which is kind of the I guess the balance or the difficulty in the whole field, which is not good for biology, right? Because if we don't develop deeper conceptual understanding in how we handle this, I mean, we're currently facing this coronavirus crisis, which is really a disaster on the human population. And we want to have quick measures, a deep understanding. And so there are many, many challenges, right? And it probably is futile to accumulate more and more data, not being able to process it appropriately. So the challenge is there.
I guess one of the few things that's cheered me up in the last few years is we started a new undergraduate sort of biology major at USC, quantitative biology. And we've, I guess, had 3 or 4 years of it now. And these kids take the computer science courses that our computer science department weeds out their majors with. They take serious prob stat, they take biology, and they're so keen. They're smart and keen, and they're really engaging these problems. I think that's a generation that will have a big impact. People that get seriously multidisciplinary education, not just trivial put your toe in, but some serious work in different areas. So anyway, it's a really complicated pathway ahead of us, and I doubt that I can contribute much to it, but I see hope there.
Well, I mean, also this goes to Chris. I mean, we should actually listen and maybe try to get together with computer science now that Kevin is joining us here also. Maybe we should try to create a major like this, or so develop such a path, right? Because you're not well served with mathematicians who want to stick with mathematics, or with the mathematicians who want to apply mathematics in light of the challenges that we're facing. And if, for instance, UVA can have an innovative coursework in the course of those things, so I'd certainly be interested in discussing this further with you.
Sure.
So at the end of my little moderation here, before we get to the posing of random questions from the audience, I just want to ask you some completely unrelated things. So probably it's interesting, at least, to know, what do you do to work out? How do you stay fit, Mike?
Well, this isolation because of the virus has really changed things. And after a little while, I realized that I wasn't getting, you can't go to the gym. And so I get up about 6:15 and walk for an hour and a quarter. Initially, you couldn't buy weights when this first happened. So I filled up some milk jugs and used them for weights. But now I've got some more sophisticated ones. So pretty much long walks and a little bit of exercise.
But prior to this, you actually used the gym and stuff, and you have like a pretty organized routine, right? That you execute.
Yeah, definitely. Don't have that, it's not good.
So soon to be longer hikes in the outdoors once this has all calmed down.
I hope so.
Right. All right. So with that, I would ask Paul to open, or Grace, who can get the questions from audience, if there are any questions for the talk.
Yeah, we can open it up to questions from the audience. Just unmute yourself if you have a question, and we'll go from there.
I have a question. You made mention that they don't know even today what the actual value of the alpha constant is for the longest common subsequence problem.
Right.
Do you know of any progress that has been made? Are there any bounds on it?
Oh, there are bounds. There were, I think, maybe even Svadl and Sankoff had some bounds. In the 80s, we worked hard on this at USC, and it was an effort to trying to improve the upper and lower bounds. And I threatened to make a plot of what my colleagues' conjectures for alpha was over time. Yeah, and so there's no tight bounds on it.
But the value that you quote, the 0.82, or
yeah, right. There are some. I can't tell you the best number there, but there's a known best upper and lower bound.
Okay.
But it's around that 0.82.
Okay.
Which to me is kind of astonishing. You go from a situation where it would be half the things matching, and then you allow these spaces to go in, and it boosts it by over 50%. And the reason so here's kind of a neat issue. You could see me implying that we'd made serious progress on local alignment statistical distribution, and not much progress on global alignment statistical distribution. Why? And my intuitive explanation, not intuitive, my explanation for that is on local alignment, the longest exact match or something, there's probably one of them. You can get your hands on its character. But if you look at the number of global alignments in longest common subsequence, it grows exponentially. There are huge numbers, and even the distance between, if you did a two-dimensional plot from 0, 0 to NN, the sort of the upper envelope and the lower envelope, I've forgotten, it's like square root n or something. There's a tangle of paths through there, and you can't get your hands, at least we couldn't get our hands on a precise statistical description of what those paths look like.
Okay, I see.
And for the variance conjecture, though, the progress that has been made was to bias one of the sequences to try to get it really in some way try to get their hands on they were restricting the distribution to be able to get their hands on some of this description I'm talking about. So anyway, it's an interesting area. There's a problem called the longest increasing subsequence of a random permutation. You take 1 through n and you permute them randomly, and then how do you go like from 2 to 7 to 19? So and that distribution it sort of looks equivalently hard to longest common subsequence, but there's some very deep work that has an exact distribution for the length of the longest increasing subsequence. And people, Aldous, Percy Diaconis, were all optimistic that that problem being solved, then the longest common subsequence problem would happen. And I hope to see it someday. It definitely won't be me, but I would really like to see this problem done.
You're talking about the Young tableau, so to speak,
Yeah,
the setting of Young tableau.
Yeah, exactly. That's definitely
the K non-crossing.
Yeah.
Yeah, so there's some whale chambers and reflection principles involved.
Yeah.
So that's very interesting stuff. So it's actually, yes. I mean, the problem is exactly analogous to that of deriving these Cannon crossing arc diagrams.
Yeah.
Because this is a bijection between these arc diagrams and sequences of young tuples. So this is fundamental work of Richard Stanley.
Yeah.
But not ad hoc. I'm kind of wondering about something else. So when we look at this genetic data, you already brought up that actually for the height, right, you have these multiple factors that play a role here. Like you had like 700 different genes and probably more, and they're not in a simple way orchestrating the resulting height,
no.
but in a complex network shape. And I want to basically say, well, if we look at the genetic data and if we do an alignment, global or local, we basically say, well, it's different, or is it a gap? And we're not really looking at the full polymorphism. So if you fix the site, we're not saying whether the sequence is different, all 3 or 4 nucleotides when we arrange them. So you always think about pairs, and we say different or not different, and then we specify potentially the difference. But we're not interested in, say, fixed site, and then whether there's an A, a G, a T, and a U for 4 different sequences, for instance, and map these events. So we're not thinking about triangles or tetrahedra. We always think about edges when we run our alignments. And I'm kind of wondering whether are you aware that something like that has been done to kind of, get to a different level of resolution in terms of interpreting those data?
Well, of course, as you know, people look at multiple alignments.
Sure.
And try to infer structure from that. And the discovery or whatever establishment of archaebacteria, this kingdom that look like they would be E. coli, but are more closely related to us than they are E. coli. That was discovered by Carl Woese and his colleagues by looking at ribosomal RNA structures from different species and relating not only the letters in the multiple alignment, but the structure. The second, that was pretty much, Woese and Fox started with 5S, but 16S was really, had enough complication that you could push this forward. So they were looking at what was in helices, what was in loops, and what were the compensating changes. So there's a very complicated look, but again, down at the pure sequence level.
Yes. I mean, just talking about pure on sequence level, right? I mean, if you take 2 sequences, say, fix a position, you have an A, you have a U, well, you count this to be different. But if you find a third sequence where you have an A, U, and then a T, say, then you look at the 3 and you say, well, look, right? I mean, they differ in a different way than the 2 respective differences. And I guess that's what I'm just reflecting upon, whether we could take advantage of these kind of thoughts. So do we have any more questions for Mike? I guess the talk was very clear, Mike. So let me take the opportunity.
There's more than one possibility
there. So let me take the opportunity to thank you for your talk. And I will probably hand the virtual microphone to Chris Barrett, and who's going to give us closing remarks. And I will say that we're going to meet again. I think the links have been posted by Grace for this town hall at 3 p.m. And so I want to encourage everybody to participate in this. And so we reconvene at 3, where you have an opportunity to, chat with Mike about anything and everything you want. So thanks from my end, and I hand the microphone to Chris.
While we're waiting for Chris, Christian, just to clarify, I didn't put the link for the 3 p.m. in the chat. If people are interested, just so we can control numbers and know how many people are coming, if you could shoot me an email, I put my email in the chat. I'll do it again so that it's front and center for everybody. But if you send me an email, I'll forward the Outlook invite that has the Zoom link for the 3 p.m. informal talk.
And I did see another question pop up somewhere on my screen, so I don't want anybody to be not heard.
Sure, I can read it. Is a question related to multiple sequence alignments. Here are some algorithms such as profile HMM. What are the main factors we should consider when we design score function or algorithms? So I can go back to that local alignment algorithm, and the relationship between scoring and what you find is, of course, critical. If you give a 1 for matching an A with an A, and you put minus infinity for everything else, your best local alignment will be a string of A's. But if you put 1 for A's matching G's and C's and T's, then you'll get a mixture. So what these algorithms find is related to what you're looking for. There's a connection to the Neyman-Pearson lemma on looking at ratios of likelihoods. So at any rate, there's an intimate connection between your scoring parameters and what you're going to be efficient at finding. And there's some kind of deep work about the matching statistics that makes that pretty explicit. So I hope that answers. The answer is pretty simple. You find what you're looking for. It's just being able to interpret your scoring parameters as being what you're looking for is the tricky part.
So hi, Professor. Yeah, this I just asked a question here. Sorry, so late. And yeah, following the same question is, when we do multiple sequence alignment, for example, some sequence will match and some sequence won't match at specific positions. So in that case, we have to allocate specific such as sorry.
So am I on, Grace?
You are. There's one last question though, Chris. So as soon as that's finished,
Okay.
we can do closing remarks unless there are more questions. Go ahead with your question.
Okay. Yeah. Just
am I on now?
There's just one last question.
I don't know what to do. There's something is wrong.
hello.
Yes, there's something wrong with my link.
I'm not hearing.
Go ahead with your question, and I'll touch base with Chris.
Okay. So my
Sorry for
question
that.
is, yeah. My question is here is when we do multiple sequence alignments, more than 2 or even hundreds of sequence, when we try to align them, then maybe some at specific positions, maybe some sequence will align correctly, but others may be mismatched. So how will we allocate the scores for each position, or for each nucleotide, or for each base?
Yikes. Yeah, I'm not sure I know the answer to that. When we were first doing this in our first paper, we just put a function for the for the multiple alignment, but the interpret you can kind of do an interpretation like I was talking about with pairwise alignments, but I think it's not as meaningful when you're looking at the 100 sequences lined up. So I'm not giving you a good answer, and that's because I really don't have one.
Okay, sure. Thank you. Thank you for your answer.
Chris, if you're on, if you want to close us out, but if you've got a problem with your link, we'll just give it a second and see if you're able to join. There we go.
I apologize. I have no idea what's going on here. It's embarrassing. Thank you very much, Michael. It was wonderful. I don't even, every time we have a conversation like this, I always leave with, well, a lot of questions, but and a lot of sort of exciting, just sort of ill-formed ideas running around. So I really hope that this afternoon that you can find some time to have some enjoyable interaction on a less sort of organized basis, and we're really looking forward for going forward. I think that this notion of sequences is not going to go away anytime soon. I've got some kind of strange background noise going on here. It's very distracting. I'm not sure what it is, and I'm going to mute myself and just say thank you very much and leave it at that. I apologize very much for whatever is happening here. I don't know what to do about it. Bye, Michael.
Oh, right. Take care.
Thanks for having me. I wish we were all in the same physical location, but that's what it is.
We will be soon, so everybody gets their shots, and then we're gonna have you for a Blackboard talk. I'm definitely looking forward to that. So that is something very nice, and to welcome you here finally at the Biocomplexity Institute. So it has been a very difficult time with all the insulation. All right, so I guess we're gonna meet at 3, right? And Grace, you kind of organize the links and stuff. And if we have to, we have multiple sessions, but you just keep me posted on this one. And I want to thank everybody for joining us here, right? It was a privilege to have you, Mike, and we're gonna have you soon in the afternoon session. So take care, guys. Goodbye for now.
Everybody.
Thank you.
And thanks for joining.